And salutations, ladies and gentlemen. My name is Andrew Williams, and this past summer, I've had the overwhelming prestige to work under the mentorship of Dr. Kar Muthmani with the assistance of Saliki Flingai at the University of Pennsylvania, Perelman School of Medicine, Department of Pathology and Laboratory Medicine, on a project entitled Characterization of Novel Monoclonal Antibodies Against Dengue Virus. Now, before I delve into the specifics of my research, I would first like to provide you all some background information so you can further your understanding of my experiment. Dengue fever is a viral disease transmitted by Aedes mosquitoes. It varies in severity from its most common form, which is not very harmful, to dengue hemorrhagic fever and dengue shock syndrome, both of which could be life-threatening. The more severe forms of dengue fever are caused by antibody-dependent enhancement, also known as ADE. The phenomenon of ADE may be observed when the patient who has been previously infected with dengue virus is infected many months or years later with a different serotype. In such cases, the clinical course of the disease can be fatal, and these patients have higher amounts of viremia. It is estimated that around 400 million people are infected with dengue fever each year, and is prevalent in many densely populated areas such as South America, Africa, Southeast Asia, and Mexico. Dengue virus is a single positive-stranded RNA flavivirus, and is the cause of dengue fever. There are 5 antigenetically different serotypes. Infection with one serotype induces the production of neutralizing homotypic antibodies against that serotype and provides lifelong immunity. The antibodies produced dengue virus also induces some sort of cross-protective immunity against the other 4 serotypes, but this only lasts for several months to a year. The difference between each serotype lies within their envelope protein, as they only portray a 65% similarity ratio. The virus has a genome of 11,000 bases, which encodes for a single large polyprotein that is cleaved into 3 structural proteins, 7 non-structural proteins, and short non-coding regions on both the 5' and 3' ends, all of which are necessary for viral replication. Viral replication is the formation of biological viruses during the infection process of the target host cell. During viral replication, a virus binds to the host cell and injects its genome into it. The genome then replicates using the host cellular machinery. Viral components and enzymes are produced and assembled, and the newly produced viruses are expelled from the host cell. However, dengue virus can also perform this in monocytes and macrophages. It hijacks the cell through the usage of its antibodies and injects its genome into it. This makes it particularly hard to develop DNA vaccines against it. By preventing it from binding, it could also prevent ADE. Antibodies, also known as immunoglobulin, are large Y-shaped proteins that are used by the immune system to identify and neutralize pathogens. There are 5 types of immunoglobulin, but my research specifically focused on immunoglobulin G, which are produced by humans and other non-human primates. Monoclonal antibodies are monospecific antibodies that are identical immune cells that are all clones of a unique parent cell. They're exemplary options for DNA vaccines. Antibodies are also composed of 2 major regions, the FC and FAB region. The FAB region is the part of an antibody that binds to an antigen. The FC region is the part of an antibody that interacts with cell surface receptors, also known as FC receptors. All the FC gamma region receptors belong to the immunoglobulin superfamily, are the most important FC receptors for inducing phagocytosis of opsonized microbes. This family includes several members, which differ in their antibody affinities due to their different molecular structures. So essentially, what we wanted to accomplish was for our monoclonal antibody construct to bind to the target pathogen, that being dengue virus, while also protecting the monocyte or macrophage by having an FC gamma region mutation that stops interaction with the cell surface. This leads me to my research design. The purpose of my experiment was to demonstrate that monoclonal antibodies could express in vitro and in vivo and determine if they can protect against virus-only infection and ADE lethal infections of dengue virus serotypes. We hypothesized that DNA plasmids encoding an anti-dengue virus human IgG neutralizing antibody with the Fc gamma region mutation that prohibits cell surface interaction will express in vitro and in vivo in mice. So the first step for my in vitro methodology was to transfect the anti-dengue virus DNA plasmids in 293T kidney cells and collect their supernames. The supernames were then purified, concentrated, and approximately quantified using Bradford assays. Using the remainder of the supernames, a few quantification ELISAs were ran to determine the exact amount of antibody being expressed. For the in vivo methodology, we first injected HC3H mice with a DNA plasmid and then bled them according to a blood collection schedule. Using the serum collected from each mice, another quantification ELISA was ran to determine the exact amount of antibody being expressed. Here are a list of my materials, all of which were indispensable to the completion of my experiment. Transfecting the anti-dengue virus DNA plasmids took 4 days to accomplish. On the first day, the existing media in the T25 flask was discarded and rinsed with trypsin. Then we took 500,000 to 700,000 of those cells and plated them in 5 petri dishes. The plates were then left to incubate overnight. During the second day, 5 Falcon tubes were filled with 300 microliters of DMEM, 60 microliters of turbofectin 8.0, and 5 microliters of our DNA plasmid constructs, shown here. This is how the constructs were named and how they are characterized. We then added the constructs to each plate and left it to incubate for 48 hours. After the incubation period was over, the plates were removed and then aliquoted into labeled Eppendorf tubes. For the purification and concentration, we had to assemble the PROCEP A media spin column first and equilibrate it with binding buffer A. While this was occurring, the sample was pre-filtered through a 0.22 micrometer Sterafluid filter device to remove any debris. The filter sample was then diluted at a 1-to-1 ratio with buffer A and was spun in a centrifuge. Then we washed the spin column with buffer A. The eluted bound IgG, we then eluted the bound IgG with elution buffer B and directly into a centrifuge tube that contained neutralization buffer C to bring it down to neutral pH. The PROCEP A media column was then washed with elution buffer B2 and re-equilibrated with buffer A. And then we desalted and concentrated the remaining antibody proportion. So for our Bradford assay, initially the dye region was prepared by diluting a dye region concentration at a 1-to-4 ratio with double deionized water. It was then filtered through a millipore filter device. 5 dilutions of a protein standard were prepared, which were representative of the protein solution to be tested. 10 microliters of each standard and sample solution were pipetted into separate micrometer plate wells, and 200 microliters of diluted dye reagent was added to each well. It then incubated at room temperature for 5 minutes and was read at an absorbance of 600 nanometers. So for our ELISA, we first made a dilution of Bethel goat anti-human Fc IgG purified and PBS and added it to each well and left it to incubate overnight at 4 degrees Celsius. The following day, the plates were washed, then PBS plus 10% FBS was added to each well and left to sit at room temperature for an hour. After the plate finished incubating, it was washed again, and a dilution of supernatant and PBST plus 1% FBS was added to each well and left to sit for another hour. After this hour was completed, a dilution of one of our 2 secondary antibodies was added to each well respectively and left to sit for a third hour. Once the incubation period was over, the plate was washed and detection substrate was added to each well and left to sit for 10 minutes. After the 10 minutes was over, we then added stopping solution, sulfuric acid to stop the reaction. And then the plates were read at 400 nanometers. For our mice vaccination, we injected each mouse with one of our 2 DNA plasmid monoclonal antibody constructs that were used in my in vitro portion of my experiment. The reason the C3H mice strand was used was because they can easily be used for dengue mice challenge studies. 4 mice were vaccinated with KD1P44A monoclonal antibodies, while the other 4 were vaccinated with LD3P48 lambda monoclonal antibodies. The mice were then quickly electroporated in the same area of the vaccination to increase the permeability of the muscle cells' membranes, allowing the DNA vaccine construct to be introduced into the cells. Blood was collected from each mouse according to a blood collection schedule, once on day 0 when the mice were vaccinated, and once again 5 days later. After the blood was collected, it was spun down in a centrifuge, and the resulting serum from the blood was used for a quantification ELISA. This brings me to my results. So the table to the right was derived from our Radford assay results, and we used it to create the standard curve. And at an OD value of 600, this is what our standard curve looked like. However, the LD3 Bradford assay, the standard curve was a bit higher than the Kappa light chain Bradford assay. And then the standard curve to the left was used to create this bar graph. There was some expression showed for the KD1 monoclonal antibodies, but it was not as much to be expected. However, the lambda light chain quantification ELISA showed much more expression in the in vitro portion. This graph represents the amount of human IgG that was detected in the in vivo mice quantification ELISA, once on day 0 and on day 5. And as expected, there's no expression shown on day 0. However, for the KD1P4 and the LD3P4, there's around 150 nanograms per milliliter of expression being shown. And it is expected that the PVAX vector showed no expression on both day 0 and day 5, as it acted as our negative control. This leads me to my discussion. To reiterate my hypothesis, we hypothesized that DNA plasmids encoding anti-dengue virus, human IgG neutralizing antibody, and the Fc gamma region mutation that prohibits cell surface interaction will express in vitro and in vivo in mice. This hypothesis was accepted since there was an ample amount of human IgG detected in both the in vitro cell culture quantification ELISA and the C3H mice quantification ELISA. As for future research, we would be ecstatic to see if the monoclonal antibody constructs expressed in vivo in mice could also bind to dengue virus serotypes in vivo as well. Testing many other novel monoclonal antibodies that affects dengue virus and conducting dengue virus mice challenge studies is also of interest. Here are a list of people I would like to acknowledge as core components in the completion of my experiment. Without these figures, the completion of my experiment would not have been possible. Thank you for your time, and I would now like to open the floor to any questions, comments, or constructive criticism. Yes?
Maybe I missed it. Why did you choose day 5 to measure the production? Why not, for example, day 2 or day 3?
Day 5? Well, we had time constraints, and it was at the end of my stay, so I wanted to see the most amount of difference that I could from day 0.
Could have missed it from day 3, right? Maybe it declined after a certain time.
Possibly. Thank you. Yes?
So one of the weird things at my age, when we brought up a lot called antibodies, right? They're kind of funny because they reproduce what happens in vivo, and that's basically an accident. You hope that a clone will amplify and does what you want, you select those clones. But now, of course, we can choose PJ sequences and take ones out of your cells, right?
Yes.
And make a molecule. I guess we could still call it an antibody, but we have a lot more control over it than what nature provides, right? So have you thought about that at all? I mean, do you think that the antibody that you produce, because you actually, if I understood what you said, you need an affinity and a disaffinity. You need the affinity for the viral protein, and you need a lack of affinity for the cell protein. Yes?
That was for the Fc gamma region.
Right. So then of course, there's a whole lot of biology known for the gamma regions, in the sense that you pick a domain, you pick a protein sequence, stick it in there, and optimize it. The part that's going on to the virus, evolution gave you one, but you can make more. Right, so have you thought about that at all? Do you think is it possible, do you think, to take that sequence and evolve it into something that might be even better than what came out of monoclonal antibody production?
That's the dream, yeah.
There are companies out doing that. There are companies. I have one friend who works for a company that doesn't use mice or cells. Heck, why rely on God when we can do better? No, they actually design their antibodies in a sequencer.
Well, we got our constructs from a lab that we work with in California.
Right.
So this is what they gave us.
Yeah, but I'm just thinking, you're a guy thinking ahead. How much of this is going to be stuff that we still rely on what evolves in a culture dish or a mouse versus how much we get smart enough to make on our own. That's all. Yeah, the other thing about it is that so your thing is going to depend upon how well this works clinically, right? It's going to depend upon how do you see this being used clinically? This is an antibody, so it's only going to last as long as you put it into the patient, correct?
Yes.
So it's good for treating somebody with a viral load, but it won't treat somebody who doesn't have the viral load and you don't want to prevent them from being infected. So would it be better if vector sources had a vaccine rather than at this time?
Yes, we wanted to create a DNA vaccine that could treat all 5 serotypes of dengue virus.